joining us. So on the top right-hand corner of your Zoom, there's View. You can click Gallery so that we all remain in Gallery. Does that work for you?
My recording webinar is now streaming. Gallery. Raise hand, record.
It's on the right-hand corner of the black square.
Yeah,
The
there's
top right-hand.
View, open up View.
Yeah.
If you click over it, if you open it, it has three options: speaker, gallery, full screen. So click.
Click Gallery.
Yeah.
You're right.
There you go.
Awesome.
Yeah.
So then I'll say a few words of welcome. I'll pass it to Paula, who will pick up from where I left off. She'll introduce you both. And then we're going to have you both spotlighted. So Lauren and Shimon, you'll be on the screen the entire time. That's what the audience will see. That's what we'll have for the recording. If that's good with you. We, excuse me, are very much looking forward to your conversation and can't wait, really can't wait to get cracking. So here we are. It's noon, Doug.
Yeah, I'm going to start the webinar, and in a minute I'll pass it over to you, Anna, and we'll get started.
Sounds good.
Webinar started, and over to you, Anna.
All right. Good afternoon. Welcome in, everyone. On behalf of Fourth Space, nous aimerions commencer par reconnaître que l'Université Concordia est située en territoire autochtone, lequel n'a jamais été cédé. Nous reconnaissons la nation Kanien'kehá:ka comme gardienne des terres et des eaux sur lesquelles nous nous réunissons aujourd'hui. Tiohtià:ke, ou Montréal, est historiquement connu comme un lieu de rassemblement pour de nombreuses Premières Nations et aujourd'hui une population autochtone diversifiée, ainsi que d'autres peuples y résident. C'est dans le respect des liens avec le passé, le présent et l'avenir que nous reconnaissons les relations continues entre les peuples autochtones et autres personnes de la communauté montréalaise. Fourth Space is activated daily via live events such as this one to create engagement around the various initiatives and research projects happening across Concordia University. It's therefore our pleasure to collaborate with our colleagues in the Office of the VP Research and Graduate Studies to host the first in a series of conversations between Concordia Distinguished University research professors and their colleagues. We are really thrilled to welcome you all in for this live dialogue recording, which will, in due course, be released as a podcast across our channels. We are live streaming this event, and I'll share that link in the chat now-ish. And of course, we've also reserved a few minutes at the end for your questions and comments, so do feel free to jump in. All right, to launch the conversation series, it's my great pleasure to pass the floor to Paula Wood-Adams. Welcome, Paula.
Thanks, Anna. My name is Paula Wood-Adams, and I'm the Interim Vice-President for Research and Graduate Studies here at Concordia. With research excellence a hallmark at Concordia, it's no surprise that our faculty members are among the most celebrated researchers in their fields. From new discoveries to the continued expansion of knowledge in existing areas, our researchers are known locally, nationally, and internationally for their work. And while there are various research awards and accolades at Concordia, the title of Distinguished University Research Professor is unique. This annual distinction is Concordia's most prestigious form of recognition for overall research achievements. It recognizes faculty who have demonstrated outstanding and sustained distinction in research over the course of their careers. They must be recognized nationally and internationally for both impact and leadership in their field. Since the award began in 2018, only 8 researchers have been awarded this milestone title. Shimon Amir, a professor and honorary Concordia University research chair in psychology, was one of the first 2 recipients of this honor. His list of achievements is lengthy, and today we will hear him discuss his outstanding career and notable research with Lauren Segall. She's a former student of Shimon's and is the current advisor for research development at the Gina Cody School of Engineering. At this point, I'd like to pass the baton over to Lauren and Shimon.
All right.
Thank you, Paula.
Thank
Hi,
you,
Shimmy.
Hi. Hi, Lauren.
How are you?
Good.
Thank you for asking.
Very
me.
good. Yeah. Well, one
Thank
of my favorite students ever.
Oh, good, because that was one of my questions.
Yeah.
Who was your favorite student? So we know.
One
now
of
we've
them.
gotten that
One
out
of
the
them.
way.
One of the most favorite students. Yeah.
Yeah, be careful, because I think there's other students listening. Thank you for asking me to be your interviewer, and it's an honor, and it was such a pleasure to be a student in your lab, and it was my great good fortune to end up in your lab. As you know, it was a bit of an accident.
Yes.
And I think I probably stayed there for about 2 years longer than I should have, because I just didn't want to leave. So it's hard to know where to start when you have such a long and illustrious career, such as you do. But I thought I would take you and the listeners and viewers on a bit of a journey over the course of the events and decisions and landmark discoveries that set you on the path to where we are right now. So without further ado, we'll get started. You did your PhD at McGill and your postdoc at Concordia.
Yes.
In neuroscience, and you had early success primarily in the field of addiction, where you were looking at opioid use and alcohol. And then people probably don't know this about you, but then you went and did clinical psychology and were a practicing clinician for several years before coming back to
Part-time, yeah.
Before coming back to Montreal to accept a tenure track position in the newly established Center for Studies in Behavioral Neurobiology, or the CSBN.
Correct.
So the first question is, why research? Why did you go into research? Why neuroscience? And then why did you come back to it?
So research, I think it was all because I had a great mentor for my master's studies in Tel Aviv University, Professor Shimon Amir, who also is a former faculty member at Concordia. We happened to meet in Tel Aviv while I was starting my master's in clinical psychology, and he really seduced me into doing research, and the rest is history. I got caught by his passion for science, for research, and for discovery, and the process of discovery. And it took me in a storm, and this is, I guess, when it all began. Because as an undergraduate student, I wasn't that interested in research. I was more inclined to do clinical work, but Zalman, as a clinician and a researcher, showed me the way. And this is when it happened, when it started. And that's what I've been doing now for the last, I would say, 50 years.
Mm-hmm.
Yeah.
So would you say that working as a clinician informed the direction of your research initially?
Not really. I did my master's in clinical psychology, but the thesis was an experimental thesis using animal models to study. I started to look into alcohol addiction as a master's student already under Zalman's mentorship. And the clinical aspect was a sideline that I thought I would be good at. Turned out that I was okay, but it didn't give me what I really looked for in academia. So research became the main theme of my involvement in this world. I moved to Montreal to do a PhD in psychology at McGill, and this is where I shifted completely to research. after I completed my postdoctoral fellowship at Concordia, the Center for Studies in, well, it was Center for Studies in Alcohol. No, I already forgot the name of that center that came before the CSBN.
I don't remember either.
Center for Studies in Drug Addiction, I remember, or anyways, as a postdoc, as a PhD student, I worked on alcohol addiction and discovered the role of enzymes of alcohol metabolism in the brain on the propensity of rats to consume alcohol, the role of these enzymes. When I came to Concordia to do a PhD, I shifted from studying alcohol addiction to address questions about the role of endogenous opiates. This is the time when endogenous opiates were discovered, and I very quickly shifted to that area because it was so curious, it grabbed me very hard. So I started to study the role of endogenous opiates and stress in various behaviors, particularly analgesia or pain responsiveness in animals. When I finished my PhD, I just wanted to finish to get back to the clinical aspect. When I came back to Israel and went to work at the Weizmann Institute, I did research, but in addition, I completed my internships in clinical psychology and actually did some clinical work as I was going on. But I discovered that while I was doing clinical work, I was thinking about science. So I kind of like left that and focused on science only.
Why Montreal? Because you seem to come back to Montreal. What was the neuroscience research landscape in Montreal?
Well, the first time I came to Montreal was because Zalman, who also was a former graduate student at McGill, suggested that I'll go to McGill to do a PhD. Under his mentor, Dr. Muriel Stern, who passed away a long time ago. I went back to Israel for 8 years, worked at the Weizmann Institute, and after 8 years, I, for various reasons, moved back to Montreal and got this position at Concordia. And that was in 1987. So I've been at Concordia now for nearly 35 years.
And was there innovative work going on? You were surrounded by a group of researchers in the CSBN doing interesting work. Montreal was a rich landscape for collaboration as well.
Right. It was a really exciting time. And I mean, this was kind of the beginning of CSBN. There were only 4 members of CSBN when I joined. I became the 5th member. Zalman Amir, Peter Shizgal, Jane Stewart, and Roy Wise. I was the 5th one to join CSBN. And it was an honor and very exciting to me. And that's kind of started my career at Concordia.
Okay, before we go any further, you are a world-renowned, internationally renowned researcher in the field of circadian biology. So before we go any further, can you just give a brief description or explanation for those listening who are not familiar with the concept, what circadian rhythms are and why they're important?
So circadian rhythms are those daily oscillations or fluctuations in a host of physiological, metabolic, and behavioral processes that enable animals to, or humans, organisms, because circadian rhythms are found in all levels of biology, allow organism to adjust well to their external environment, primarily to the solar cycle. And so, so those are the rhythms. This is what we can see with our eyes. or measure in animals. And these rhythms are not a direct response to the environment, but rather they're driven internally by clocks that are called circadian clocks. These clocks drive the rhythms, and they are synchronized with the external environment, primarily via the light-dark cycle. These clocks, should I continue to tell you what these clocks are based on, or?
Well, we'll get into it.
Okay.
We'll get into it.
Okay.
Just a brief introduction for so that
I just want to add that they're crucial for normal adaptation of
right.
organisms to the environment.
Right, and they're adaptive, and if
Yeah.
they're disrupted.
So disruption of circadian rhythms lead to a host of disturbances and pathologies. So they're really, really fundamental to biological regulation and survival.
Yes. So the course of research is rarely a straight line. You're in circadian research now. But I remember when I was a student, just to paraphrase you, and I think actually you were paraphrasing B.F. Skinner, but you said to me once that in the course of a line of inquiry, if something unexpected crops up, that you should drop the first line of inquiry and run in the direction of the unexpected discovery. So what was the discovery that pointed you towards circadian research? Because we saw you didn't start there.
Yeah.
And what kept you running in that direction? What was the discovery in your recollection? And why was it important?
That's kind of tough to pinpoint. There was something that kind of led me to focus on circadian biology, because prior to that, I kind of shifted. I moved from area to area because I was just seduced by science to the extent that I made a finding, I made a discovery, I published the results, and then all of a sudden I found something new, and I kind of like got excited about that. And I think that the finding that got me going in a serious way in circadian rhythm was it was kind of like almost unintended finding. It was a thought that circadian rhythms can be, well, we knew that circadian rhythms are synchronized with the environment via the light-dark cycle. But for whatever reason, I came up with the idea that instead of that stimuli, events that are associated with light, with the light-dark cycle, can themselves entrain circadian rhythms. Stimuli that initially are not effective in entraining circadian rhythms, but as a result of an associative learning process, they become effective in synchronizing rhythms. That finding was published in a very good journal and
Nature.
In Nature, yeah, and allowed me to receive a major funding from CIHR to continue to study this phenomenon. And actually, this was, I think, just at the beginning when clock genes were discovered. This was clock genes are the gears of the circadian clock that drive circadian rhythms. And this was at the time when clock genes were discovered. And so there was a lot of excitement in the area, in the field, about circadian rhythms. CIHR found this unusual finding that we had in the lab and funded my research, and it escalated from there to various aspects of chronobiology and circadian rhythms and the genes that drive them, etc.
So yeah, so that was your 1996 Nature paper that really really
Right.
put you on the map and solidified your reputation also, as because I know before you were also looking at thermogenesis and nitric oxide in the SCN as signaling, but
Yeah, I mean, before that, I was very interested in thermogenesis, which is an adaptive response to cold and diet, heat production in a tissue called brown fat. And I was studying how this internal furnace of the body is responding to various external stimuli. And we found that indeed the suprachiasmatic nucleus, it's a region of the brain that harbors the master circadian clock in mammals, that the suprachiasmatic nucleus had influences over this brown fat thermogenesis. Yeah, so that was before I really moved to focusing on circadian rhythms per se.
right.
Yeah.
Okay, so the Nature paper was really
The
what sort of
paper
solidified you
Yeah.
researching circadian rhythms. So
Yeah.
you've been widely praised for bringing a really truly multidisciplinary approach to research. So as you mentioned, it was really the discovery of clock genes that were driving the circadian clock.
Yeah.
So you've been really lauded for bringing not just behavioral and neuropsychopharmacological approaches, but genetic, molecular, cellular, biochemical,
Correct.
and even genetic interference, which has also really, I think, allowed you to pursue these discoveries. So that was a seminal and pivotal moment that really changed the landscape.
And I was really blessed by having great students who really helped me accomplish all these to really go deep into research of these different issues. I wouldn't be able to do that without them, so it is these accomplishments have to be and must be shared not with the students that that came to the lab. Yeah.
So continuing on the theme of the paper, so basically what you're saying is conditioned non-photic stimuli, so environmental stimuli that wasn't light.
Yeah.
Normally light is what tells the master clock.
Yeah. Actually, there are two major synchronizing stimuli or events in the environment that can alter the phase or can synchronize circadian rhythms. Light is the most important one, but then there food intake, eating, which is also a very powerful stimulus for circadian rhythms. Although the mechanism that mediate the effect of feeding on circadian rhythm is not as clear as the one that mediates the effect of light.
Right.
But both external events have powerful effect on circadian rhythms.
Right. So subsequent to that, you were also one of the first to identify and characterize peripheral clocks within the brain.
Right.
And that was a really big thing.
Yeah.
So why is that so important, and what does it mean in terms of human pathology and human behavior?
Okay. So initially, following the discovery of clock genes, people in the field focused on clock gene expression in the suprachiasmatic nucleus. This is the region that I mentioned before that functions as the master clock that controls circadian rhythms. But once circadian clock genes were identified and shown to be critical for circadian rhythms, it became evident that these genes are expressed in all cells and tissues of the body, not only in the suprachiasmatic nucleus, which functions as a clock, but also in many other brain regions and peripheral tissues like the liver, lung, heart, etc. Initially, people weren't sure about what is the function of these clock genes, but now it turns out that there are local clocks in different regions of the brain. I'm particularly interested in the function of clock genes in the brain. So in different regions of the brain that are important for motivation, emotion regulation, addiction, etc. And through studies that enable the manipulation, genetic manipulation of clock genes within specific brain regions, it became evident that these clocks are very important for the maintenance of adaptive, normal behaviors and functions, and that in the absence of those clock genes or loss of function of these genes can lead to various perturbations in behavior and physiology. So it became very important to study the nature of these clock genes, how they're regulated, both by the master clock, because the master clock appears to control circadian rhythms in the entire organism by sending messages to those peripheral clocks. So yeah, that really attracted my attention, and we kind of moved from a suprachiasmatic nucleus-centric research to studying clock genes in different regions of the brain. And as you know, you were heavily involved in this research by discovering a very important role for stress hormones in the control of clock gene expression in stress-related brain regions. I think I
remember. So can we yet tie gene expression to behavior?
Yes, of course, we can. And we do that by targeting specific clock genes within specific brain regions and looking at behavior and see how behavior changes as a result of loss of function or suppression of clock gene expression in the brain. And this is actually, I mean, our research now is really focusing on using various genetic techniques to manipulate clock genes within specific important regions of the brain, and looking at behavior and what happens to behavior. And this has implications for, of course, for health.
Right. So going back to the implications for health, we've seen that you can actually uncouple these peripheral clocks that are responsible for various behaviors from the master clock in the SCN, and that this is maladaptive.
And this is something that most people can relate to, particularly people who travel across time zones or people who work shifts. Exposure to light at the wrong time of day, exposure to light at night, or moving across time zones results in uncoupling or dissociation between the master clock and peripheral clocks. And people who travel across time zones experience those jet lag symptoms that result from this uncoupling of the master clock, who responds to local time, and peripheral clocks that take some time to adjust to those shifts, time shifts, or working at night. So obviously it has a very important impact on health and well-being.
Right. So for example, shift workers, like people on the front line in healthcare who are going back and forth between shifts, you see like an uptick in pathologies as a result. Yeah.
And if this becomes chronic, then there are a range of disorders that can result from, and pathologies that can result from this mismatch between different clocks in the brain and periphery.
So in the last few years, as you mentioned, you're still maintaining research in circadian biology, but you've gone a little bit in a new direction. And I was looking at your recent publications, of which there are many, and it seems to be a little bit more back on mental health disorders and looking at circadian rhythms and mental health disorders. So can you just give a little bit more, elaborate?
So this is interesting because as I told you before, I started research in the area of alcohol addiction. And for many, many, many years, I haven't done any work in this field. And then a few years ago, we decided to, for several reasons, we decided to look at the role of circadian clock genes in alcohol drinking behavior and alcohol dependence. So there was some evidence from studies in humans, particularly gene association studies, that showed that circadian clock genes or polymorphism in circadian clock genes is associated with alcohol drinking and alcohol use disorder in humans. And we decided, but there was no evidence of a causal relationship between function of these genes and alcohol behavior. So with the help of a really super team of researchers in the lab, we developed animal models where a particular clock gene that is called BMAL1, which is the most important clock gene, is deleted from a particular group of cells in a region of the brain called the striatum, which is important for addiction. And we did find that deleting or knocking out this BMAL gene from cells in the striatum led to changes in alcohol consumption in mice. The exciting finding, however, was that the effect of deleting the gene from the striatum was sexually dimorphic. Namely, it led to an increase in alcohol intake in male mice, but it suppressed intake in female mice. Now, in mice, females drink much more alcohol than males. So there is a big sex differences in the natural propensity of mice to consume alcohol. Females are simply more avid drinkers of alcohol than males. This is actually the opposite in humans. In humans, males or men drink more than females. This is another robust and pervasive sex difference. There are other sex differences in humans. For example, although more men become addicted, women become addicted much faster than men. The transition from, say, social drinking to excessive drinking, uncontrolled drinking of alcohol is much faster in females than in males. Females also suffer from more health consequences from alcohol than males. Females tend to relapse after a period of abstinence faster than males. So there is a major sexual dimorphism in addiction in humans. In mice, females drink more than males. And we found that this sex difference may depend, at least in part, on the expression of this gene BMAL1 in the striatum, because when we deleted that, those sex differences disappeared as a result of suppression of intake in females and enhancement of drinking in males. So remember, there is a big sex difference. Females drink more, but when you take out the gene, they drink less, whereas males, if you take out the gene from the striatum, males drink more. So the initial sex difference disappears. And the conclusion from that was that the BMAL in the striatum confers protection from addiction to alcohol in males, but risk in females.
Does it change across the estrous cycle?
We still don't know the role of ovarian hormones in this sexual dimorphism, but in mice, it is shown that the astro cycle in mice does not affect alcohol drinking.
No.
In females, yeah, it doesn't have a major effect. But sexual dimorphism doesn't have to depend on sex hormones only. It is probably the result of some other genes on the X or Y chromosomes, which determine the sex, so biological sex. Yeah. So we don't know yet what really mediates the effect of this gene on the sexually dimorphic effect of this gene on alcohol consumption. But we're going to find out.
Yeah. I asked about the estrous cycle because if you remember, you were also the first to look at.
Right. We did look at the role of
HER2 rhythms in females across the estrous cycle.
Right. Yeah. So Jen Perrin, my former student, who's now in England.
No, she's back in Ottawa.
Oh, she's back in Ottawa. Okay.
In Ottawa.
Right. So yeah, she did some studies and showed that the estrous cycle has an effect on the expression of clock genes in some regions of the brain. Yes, we've done that. We actually looked at that too. But in terms of alcohol consumption, it is known that ovarian hormones do have an effect on alcohol consumption in females. But we still don't, not the estrous cycle, but if you manipulate, say, surgically remove the ovaries, in females, they drink less. So estrogen has a promoting effect on alcohol consumption in rodents. Huh. Yeah, but the estrous cycle itself doesn't seem to have a major effect on alcohol intake in mice. Complicated and therefore very interesting.
Yes,
and challenging.
And yeah, and made a bit of a splash. I know people were talking about this.
Yeah. Well, I mean, anything to do with alcohol addiction or alcohol use disorder is a major contributor to morbidity, to illness and mortality worldwide. I mean, it's a very serious problem. So anytime you make a discovery or an observation about what really affects alcohol drinking, gets attention. Now, we in our studies that were just published, looked at voluntary alcohol drinking in a model that doesn't really address the issues that individual people confront in addiction. Right. So now the challenge is to see whether those findings can be extended to animal models of addiction to alcohol. And that's what we're going to do next.
Okay, so that was my next question was you've come full circle. You've looked at a whole host of factors. You've looked at feeding and circadian rhythms. You've looked at regulation of dopaminergic responses. You've looked at the role of stress hormones. You've looked at the differences across the estrous cycle. You've looked at differences in circadian rhythms in aging. You've touched on so many things. Now you've come back to alcohol, and where does your research go from here? Where do you see it in the next 10 years?
So, I mean, what the program today addresses the function of clock genes in a number of important brain regions, the striatum to begin with, but also the habenula, which is another brain region that's really, really important for actually controlling dopamine release from midbrain dopamine nuclei, which is a very important neuromodulator for motivation, addiction, decision-making, etc. So we're not looking only at alcohol dependence, but we're also looking using animal models at the role of these clock genes in anxiety-like behaviors, depression-like behaviors, etc., motor functioning. This is really the plan going forward. In fact, we're also looking at the role of clock genes in cardiovascular functioning. This is done in collaboration with a colleague, with Andreas Bergdahl and Konrad Schottner, who is a research associate in my lab. This is a new line of research that I've never been involved in, but looking at links between the heart and the brain, and how disruption of circadian clock genes in the heart can contribute to depressive-like behaviors in animals, which is a known function in humans. Cardiovascular disease is associated with this comorbid with depression, for example. So we're looking into that area of science. I think we're going to make some very interesting findings there. We're actually using manipulation of clock genes as a tool to cause a heart disease in mice, and then looking at behavior that is mediated by the brain. So heart-brain connection. Yeah, but this is really the way going forward, to look at contribution of clock genes to normal behavior and to disease. Sorry. Yeah.
So cool.
Well, it is. It is really. Yeah.
I have just a last question for you. You've already mentioned that Zalman was your inspiration and mentor. So, I was going to ask that. I mean, aside from your many achievements and recognitions and accomplishments, you have an unbelievable training record. And the quality and quantity of students that you've had are just unparalleled. And did your training and your experience, how did that influence your approach to training and take on training?
So I think I had...
Because you were amazing, by the way.
I had 4 great mentors. Zalman to begin with, but Jane Stewart and Peter Schisgall and Roy Wise were also quite influential in my work, in the way I was thinking, in writing. I learned a lot from them, and I thought I should. It was natural to pay forward, to make sure that students that come to work with me will get the best that I can give in terms of mentorship and resources and so it's kind of like that's the way it's done, and that's the way success is built on. I cannot do everything by myself. It all depends on mazel, luck, and good students and good collaborators, and that's what kind of like I think colors my career. I mean, the students, they're the ones who helped me get where I am today. I wouldn't be able to do that without students, great students. I had all my students are great. I mean, really, so I mean, first and foremost, I have to thank them for my accomplishments. Not only in terms of the work that I've done in the lab, but they taught me a lot too. I mean, it's not that I was the one who taught and they were the students. I mean, I learned a lot from my students. I learned a lot from you, particularly. Well, Lauren became my students because we had an exchange in the cafeteria in the whole building downtown. And I challenged her, and she had the chutzpah to respond. And then I think the same day or a day later, I met her on the elevator and I offered her to come to the lab, and the rest is history, right? Is that the story?
You didn't say please or thank you.
Yeah, I know.
When I was working at Java U.
Yeah.
And I told you needed to say please or thank you. It could have gone both ways.
Yeah.
I was very lucky that day.
Yeah.
But yeah, that's how we met. I always had your coffee ready, you and Jade.
Yeah. But
I was fortunate to end up in your lab. Very fortunate.
Yeah. And Lauren's papers, the work that she's done in the lab, I mean, they're very, very highly cited, by the way. I don't know if you know, but
I haven't followed in a while.
Yeah, very highly cited. So your work had huge impact. And people look at that work.
It was an exciting time.
Yeah.
And your passion for discovery.
Yeah, that's the thing. I think it's good and it's not so good, because that led me to jump initially from area to area and making contribution and move on. But my interest in chronobiology and circadian rhythms has stayed with me for quite a long time now. Yeah. And this is where I'm going to stay, I think.
Yeah.
Good.
Thank you for talking to us, Jimmy.
My pleasure.
Well, thank you, Shimi and Lauren. That was just fascinating to listen to, and it's really touching, actually, to see the strong bond between mentor and graduate student, even after a few years after finishing your degree. And I can just see that that the relationship was beneficial for both, for Shimi and for Lauren, and really also for Concordia, because we were able to hire Lauren as a research development advisor, and she continues to make contributions to this day. So your training has benefited the universities in ways that you probably didn't imagine, Shimi. We do have time for a couple of questions. If anybody in the audience would like to speak, anybody on Zoom, you can raise your electronic hand, or you can paste anything in the chat and Q&A if you'd like.
There is a question in the chat.
Oh, I see Suzanne is there.
Oh, yes. So I assume this is a question for Shimi. What advice might you give to young students today who are wondering how to get started in a career in basic science research?
Find a good mentor. Find a good lab. I mean, it's all about mentorship and relationship between students and their supervisors. There are lots of good ideas all over the place, and to take a good idea and translate it into actual findings really depends on the environment where you are. Mentorship is critical. Yeah, I think that's the way it is. People, I mean, to succeed in science, you really have to have passion for the scientific process and for discovery, and it's really exciting. But you need to team up with people who can translate your passion into actual work and findings, and read a lot, and make sure you can write. I mean, writing is critical. Reading helps writing. Those are the kind of things that will help you succeed in science.
I think both they're tied together a little bit, because Shimmy, I remember the first thing I ever wrote for you was a 1-page description of my project. I was really proud of myself.
Yeah.
And I brought it to you, and you read it, and you said, "Wow, you're a really good writer. Let's just change a couple of things."
Yeah.
The last sentence was mine. And everything else.
No,
but I learned.
Yeah, but I'm not a native English-speaking individual, and I have to say I learned a lot from my students about writing, not just from my mentors, but from my students about writing properly. You know, a scientific writing is a special way of writing, and you need to learn how to express yourself scientifically. Very important.
Well, by the end, I could bang out those papers. By the end, I could bang out those papers.
Yeah.
I really learned.
Shimmy, what do you think about the characteristics of persistence and resilience in young scientists? To me, that always seemed to be very important. What's your experience?
Very important. And, you very often, maybe even more often than not, you start a project and it doesn't work out the way you thought it will be. But I think it's very important to pay attention to why it didn't work out. And from looking deep, you can come up with new ideas. And so, you have to be resilient. You have to withstand failure because it happens all the time. Science is, again, there is a lot of luck embedded in science. Although, I mean, creative thinking and is very important, but there is also a lot of luck. But if something doesn't work, just look at why it didn't work. And if there is nothing you can say about that, just move on. I mean, there are many, many, many interesting questions to be addressed in science, endless number of questions, and, just persist. Don't give up.
I think that's what makes it such a wonderful career, is we never run out of questions. There's always something new to ask.
Right. And the scientific method is really the best method for acquiring knowledge, better than any other method that, and so, yeah. I think it's a great place to be in.
Well, I don't see any more questions coming. Lauren, did you have something final you'd like to say before we end the session?
Having been fortunate enough to play a part in just a small aspect of your career, it's my honor, and I was just so happy to conduct this interview, because I just really wanted to show you in the best light possible and do you justice, because you're an amazing scientist, but an amazing mentor. And
I just want to say once again, I really want to thank all my students who all made a significant contribution to my success, and I wouldn't be able to be where I am without them. Past students and of course current students. I have a great team of researchers working with me now, and I'm sure we're going to do some really interesting, great things.
Wonderful. Thank you. I'll pass this now to Anna for the final closing. Thank you so much, both of you.
It's a pleasure.
And thank you, Paula. We really appreciate those of you who have joined us, also braved the slippery sidewalks out here to join us in the space. And those of you who were able to join online, we're so grateful for your time. And Shimi and Lauren, it was so fun to be a fly on the wall and just feel like we're listening to this very generous exchange between the both of you, this very collegial exchange as well. We're very appreciative of your time and the moment that you came together in this way. We look forward to releasing this conversation with some love and gentle editing as a podcast so people can take a walk with it and be inspired. So on that note, we will close this webinar for you folks here today. We appreciate you. Thanks for being here. And until next time, we will be hosting a few more of these conversations with Concordia's distinguished university research professors, and we look forward to inviting you back. Congrats again, Shimon, and thank you so much, Lauren. Have a great day.
Bye.
Bye.
Bye, Lauren.